Hello, beloved Shirtloads listeners. Dr. Karl here. Welcome to some excellent repeat episodes. You see, I'm on sabbatical overseas, and I'm working on a strategy using artificial intelligence to counter climate change disinformation. Ooh, big topic. So stay tuned for updates about this exciting new project. I'll be back with new episodes in late April. Catch you then.
G'day, Dr. Karl, Shirtloads of Science, University of Sydney, talking with Professor Marcus Atlas, Professor of, now, is it otolaryngology or gology?
Gology. It's gology. Oto, ear, and laryngology, larynx and throat. And if you really want to be pedantic, it's otorhinolaryngology, rhino being the nose, of course. But I'm an oto guy. So for most of my career, I'm an oto guy. I love the ear.
And I've been reading your papers, and so the bottom line is that you think that finally you're getting really close or have cracked the ability to make an artificial eardrum?
Yeah.
Mate, it's astonishing. Like, I've been reading your stuff here. So firstly, you sent me a little PowerPoint presentation, and you're saying that hearing loss, the illness from that, CSOM, what does CSOM mean?
Infection of the middle ear with a perforation in the eardrum.
65 to 330 million, like a third of a billion, and there's only 8 billion on a planet. And you've mentioned that it accounts for 28,000 deaths and a disease burden?
It can be. There's a massive disease burden from quality of life. You can imagine the hearing loss and the ear discharge and the chronic infections. But some of those patients with chronic draining ears have got these disorders called cholesteatoma, skin growth, chronic infections of the middle ear and mastoid. And some of those can erode into the brain. They get brain abscess, meningitis, and they can die from it. So that's not common in our community, of course, but in developing parts of the world, it's a real problem. This is a real thing.
You're saying developing, but that includes wealthy people who live in cities as opposed to poor people with a different skin colour who live out of town. And I was working with Fred Hollows for a while, and he was saying that this is epidemic in Australian Indigenous communities?
Yeah, Fred was right. And of course, he managed manifestations of eye disease of difficult parts of Australia where eye disease was incredibly common. And in the same places now, First Nations people suffered terribly from chronic middle ear disease, particularly kids. And so this puts them at such disadvantage for their future, not to be able to hear, not to be able to participate, not be able to deal with language and education in the way they might want to. And so it is disproportionately affecting that part of our community.
Yeah, Fred told me, because I designed and built a machine for him to pick up electrical signals off the human retina as a biomedical engineering student. He told me that he smuggled 0.5 ton of sulfur-based antibiotics that was slightly out of date into the Northern Territory. And he said the trouble is that you go into a classroom of kids and you just hear them going snot, 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 snot. And then after the antibiotics, it got better. But he said it was just epidemic. And
Yeah.
he said that they were, is this true that if you have it for long enough as a child, you can be deaf and therefore just missing out in the community when you grow older?
Yeah, that's the problem. And not only that, but the rates of incarceration and the rates of disadvantage in adult life from childhood ear disease are huge. So it's not just a problem of childhood, it becomes a problem of adulthood and a problem of the social fabric of the community. It's a real thing. It's a real problem. And we've tried to tackle it in multiple ways. Australia has a history of trying to understand chronic middle ear disease better in First Nations people, but we haven't solved it. We haven't solved it as a problem. It's still a plague in our community. It's something that we really like to do better.
This is not an overnight problem that you can solve. And so I've been reading, you have written so many papers over the decades. So am I right in saying that where you are at now is that you have a synthetic eardrum that you can implant?
Yeah. So normally what happens when we graft an eardrum in surgery, we use the patient's own tissues. And the tissue that we primarily use is the lining over the muscle above the ear. The chewing muscle is called the temporalis muscle, and we take the lining over the top of it called temporalis fascia. We do that at the time of surgery and use that to graft the tympanic membrane. But The problem is it doesn't act like a normal eardrum. It doesn't heal like a normal eardrum. A lot of the surgery fails. So what we need is something that has specific acoustic hearing and mechanical properties to resist the problems of chronic middle ear disease. And so that's what we went about designing, and we used an incredibly unique material that I guess you'll want to talk about.
Yeah. So, by the way, for the audience, I'm sorry to say, but this is a case of where an overnight discovery takes decades. And so, my understanding, and I could be wrong, is that when you're listening to the quietest sound you can possibly hear, the eardrum goes back and forth a distance equal to the diameter of a hydrogen atom. It's also weird in how it heals. So you had to, firstly, looking at wound healing, and apparently the eardrum heals in a different way, and then understand what the heck you were trying to mimic. And so you then spent a bunch of God knows how many years on biomechanical and acoustic properties, because if you make an eardrum and it's too floppy or too stiff, mate, you missed it. And then finally, materials. I mean, you just didn't walk down the Fred's Friendly Fruitoria. So take us through the first stage of your decades-long journey in what apparently now seems to be a happy ending, the wound healing and the cytocompatibility.
To begin with, you've got to understand how the eardrum heals and how the eardrum works. And you've just described the fact that we still don't completely understand how the tympanic membrane moves and therefore transmits sound from the eardrum via the small bones of hearing, the malleus, the incus, and the stapes, to the inner ear. That's in you and me, that's in a normal eardrum. But in a diseased eardrum, it's even more complex in terms of understanding how the eardrum heals, how the eardrum behaves, and what its natural biology is. So we ended up becoming, I don't know that we ever intended it, but now we're sort of the world leaders in tympanic membrane biology, regeneration, mechanical qualities, and acoustic qualities. We just needed to understand what we were trying to achieve with our material. And a lot of it was around animal models of how does the eardrum heal, growth factors and particular factors that occur in the edge of the eardrum that creates this healing. And young surgeon scientists did their PhDs area. So we're kind of the experts in tympanic membrane healing. Our work on the acoustic and mechanical qualities we needed in the diseased eardrum to prevent it from busting again, from getting infected again, from getting retracted. Retraction means the eardrum gets drawn in, or there are weak areas. We were talking a few seconds ago before we started about weak areas of the eardrum as a consequence of middle ear disease. So these weak areas over time can get drawn in. They can cause problems in the middle ear, a disorder called cholesteatoma, hearing loss, instability. So we had to deal with all these weak areas as well as these busted up eardrums. So what do we need to create to prevent those from occurring? So that's mechanical. And then we went around material science, and that's when we hooked up with Deakin University and the Institute for Frontier Materials. They've just been wonderful collaborators, and happening upon silk.
Silk?
Yes, silk is the material that we use.
Well,
it's made out of silkworms. Yeah.
One of your papers I read, the eardrum heals in a completely opposite way to the rest of the body. And can you assume that our audience doesn't have any medical training? Can you explain how it's different in the concept of keratinocytes?
Keratinocytes are skin cells. The eardrum heals in this really weird way. So remember, the eardrum's surrounded by air.
Oh, there's air on both sides.
There's air on both sides. There's air in the ear canal, there's the hole in the eardrum, and there's air in the middle ear. It has to find a way to bridge this gap. So if we've got a cut on our hand, the skin is healing over soft tissue. There's the underlying fat and subdermal tissue, the keratinocytes, the little skin cells will migrate across and join the wound on the cut on our hand. But in the ear, it doesn't have anything to grow over the top of. So it creates this bridge. The keratinocytes grow over the top of each other and they crawl across like the Sydney Harbour Bridge, and they try to meet in the middle as they create a mound of keratinocytes that tries to meet in the middle and create a bridge.
Is that kind of like the mental image you have of ants trying to cross a stream, and they sort of build a bridge over each other's bodies.
Yeah, that's exactly it.
Wow.
And so the keratinocytes are doing an ant impersonation and coming across and slowly starting to heal. And if during that time you get a cold or you blow your ear hard, it'll bust through those thin layer of keratinocytes and it'll have to start again. The ants will have to, you know how they retreat and then start again. That is part of the problem of tympanic membrane healing. It's really unique because of the air-containing hole. We learned to grow keratinocytes. So we're the only group in the world who knew how to grow tympanic membrane keratinocytes, skin cells. And we had these colonies of human tympanic membrane keratinocytes that we could use to experiment with. We took them from patients who gave us permission to use their skin of the ear canal, and we would create tympanic membrane keratinocytes, and that's where we did all our experiments about healing over these membranes. You know, we're the guys that people come to if they need a tympanic membrane keratinocyte culture.
Wow, I never realised that the healing on the eardrum was different. And so if somebody gets an infection while they've got a perforated eardrum, the keratinocytes might give up eventually, and eventually, you know, with blowing their nose and high pressure, and then that way they would be left with a perforated eardrum and deaf in that ear forever?
Yes, permanent hole in the eardrum with an associated middle ear conductive hearing loss. So that's not unusual. And even if it heals, it can leave a weak area. And so that patient might want to pop their ear all the time because they want to tense the eardrum and improve their hearing because they've got a weak area where that hole was. Or they'll go to a weak area, and because they've got ongoing negative pressure in the ear because their eustachian tube doesn't work properly, it'll get sucked in and skin will accumulate inside that sucked in area of the eardrum. So you need a functioning, properly mechanically stable tympanic membrane.
How many years have you been going on this pathway before you end up with your final product?
Oh, do I have to tell you? It's so depressing. It's sort of like 20 years that we've been working away at this. But we've learned so much on the way.
Though, because you're doing something good for your fellow human.
We are, and we've learnt a lot, you know, because all of these things that we've been talking about are valuable not just for us, but they're valuable for tympanic membrane research. What's come from it is, as we found these growth factors that were important in healing, some of the groups in the world started to try to develop growth factor healing medicines for the tympanic membrane. They haven't worked. If I add growth factors to your eardrum when it busts, will it heal better? Will it be stronger? Will we get a better surgical result if we add these growth factors? Some of them are sort of safe to use, some of them not so safe to use, but you can see how the science, which you love so much and I do too, is not just for one thing, it's for everything. It makes a massive difference to the future of understanding the human body. So it's important.
So wound healing, weird. Biomechanical and acoustic. Okay, material development. And you're telling me silkworm?
We looked at materials and we went to our who have become great friends. Science is a great thing because you make fabulous friends in your collaborations around the world. And this collaboration with Deakin University, the Institute of Frontier Materials, has brought a group of really interesting people together, and their thing is materials. And one of the materials they're really interested in is silk and silk textiles. They've been involved in the textile industry and using silk for other types of engineering. But in this case, it was bioengineering. It was about, could we use silk, and particularly a product from silkworms called sericin, remove the sericin from the silkworm to leave something behind called silk fibroin. Sericin is a component of silk that is thought to have an inflammatory effect on healing. And so it was generally removed out of the silk to create silk fibroin, which was thought to be more biocompatible. And that's what we worked on. The guys at Deakin can turn a silkworm's silk into transparent membranes, gels, foams. It's remarkable. So it starts as silk from a silkworm and then ends up looking like ours, which is a completely transparent, micro-thin tympanic membrane graft. It's completely transparent and it has these specific acoustic and mechanical qualities that we've been able to tune according to what we think we need for a human tympanic membrane. And we base that on a whole lot of experiments on the human tympanic membrane, a cadaver tympanic membrane, to try and replicate those qualities.
Wow. Like those creatures, spiders and the silkworm, they make this amazing fabric that is weight for size stronger than steel, and yet they do it at room temperature with organic materials. How big do you get these tympanic members, which is a fancy word for eardrum? How big are they? They come out like in a sheet, a metre across, and then you stamp out little holes, or do they make little? How big do you get individual little bits?
We wanted to 3D print using silk fibroin to make a very specific shape, because our eardrum, the human tympanic membrane, it's not flat. It's conical shaped. It's like a diaphragm. It's conical shaped. In the middle is a small bone called the malleus bone. It's cone shaped from there. So what we tried to do is replicate this unusual shape, and it didn't work. We couldn't get to surgically fit it inside the eardrum. I had in my head that if we make it the right size, it'll just click into place underneath or on top of the eardrum, and it'll be so fantastic.
Underneath or on top of the damaged eardrum.
To fix the hole. And then the skin cells would heal over the top of it, and we would have a perfect drum.
And then that would, when an air pressure wave comes in, that would then move the malleus bone, and then you get the whole chain of 3 bones moving, and then you get a window moving in and out, and then you get pressure waves in the liquid, then hair cells bend over and they give off electricity, and it goes to the brain, and you turn it into hello. Okay.
You just described it.
Yeah, so you were there right at that eardrum stage.
And trying to replicate this conical shaped diaphragm that is like a microphone. Then we moved towards creating sheets. And as you say, we create sheets. They're about 8 millimeters in diameter, 60 micrometers thick, but various thicknesses according to what we need in terms of mechanical and acoustic quality. So we vary the beta sheet content of the silk fibroin. We've mixed it with a material called glycerol to improve the integrity of the drum, and then created this composite membrane which we can utilize in surgery. So it's taken ages because we've spent so much time in the laboratory using it in a surgical situation to try and find the right material that it can be used surgically, but also has these acoustic and mechanical qualities that we know we need.
How does the regular eardrum or tympanic membrane get the blood that it needs, and does your synthetic one need some sort of nourishment, or is it just set and forget?
What the scaffold does is it forms a membrane that the human keratinocytes will grow over the top of, like a bed for those ants to crawl across, something for them to crawl across to create a covering sheet of keratinocytes over the top of our scaffold. So it picks up blood supply from 2 areas. One is the periphery of the eardrum in the ear canal. That's one of the blood supplies. And the other important blood supply is coursing down the malleus bone to the middle of the eardrum. So there's blood vessels radiating out. So from the inside and from the outside
there's 2
of the
separate
drum.
sets of blood supply, keeping your normal, as given to you by evolution, eardrum, one from the outside, one from the inside. Okay, yep, got that.
They will facilitate the blood supply to the keratinocytes, and if we give them the right environment, the keratinocytes love our silk. I don't know why, but they just love growing over silk, which we showed in the laboratory. So if we can keep an uninfected, stable scaffold of the tympanic membrane, then those keratinocytes will grow. And we've shown that both in animals and in the test tube.
What you've done is not so much like putting in an artificial leg made of titanium and then you run on titanium, but rather you're putting in a substance that has good properties but is mainly a scaffold. Does it being there interfere with things further down the track?
No, it's perfect. It's perfect because it has those mechanical and acoustic qualities that we need from a drum. So it forms like what we call the middle layer of the tympanic membrane, which is generally has collagen in it. So it replicates the middle layer of collagen, and the keratinocytes grow over the top of the scaffold.
Do they grow over the top and the bottom, both sides?
No, on the inside, on the middle ear side of the tympanic, so there's 3 layers to our tympanic membrane. The outside is keratinocytes, the middle layer is collagen, a fibrous layer, and the other layer on the middle ear side, the cells of the middle ear, which are called mucosal cells. They're different.
Sort of wet, squishy ear things.
Wet squishies.
But you've got this extra membrane about, you said 60 or 80 microns thick, roughly the thickness of a human hair. It's in there forever now, isn't it?
It's in there forever, but the silk scaffold will slowly degenerate. So it's known to be a material that will slowly disappear in the human body. And we believe, we haven't been able to prove this yet, we believe that by that time the tympanic membrane will have the mechanical and acoustic qualities for long-term stability.
Oh, so how long have you been using this silk for, like 10 years?
Yeah, we have, but our experiments in animals only go out to a year. So we know the membrane is still present in some form at a year, but the eardrum has regenerated by that time.
So while you've got short-term, like 1 year animal, well, what do you have for human results?
We're not allowed to do our first human clinical trials until we've got FDA approval. And so we're in the middle of FDA approval.
Food and Drug Administration, is it FDA? The American one?
The American one, and the Australian one is the TGA. Because America is going to, of course, be a bigger market, a bigger place of requirement, we're going to the States first. And that's kind of how startups do things now.
TGA is Therapeutic Goods Administration. So where are you with the TGA for getting their approval?
We're doing FDA first. The TGA will be influenced by the FDA in terms of approval process, whereas the other way around is a bit more complex. That's what we elected to do.
FDA approval, is that 1 hour away or a month or a year or a decade, or you don't know?
It's not a decade, but it's tough. The requirements are onerous. I think it's important to know as a community that it's not easy to get a new material or a new device or a new drug to be used. And that's what we would want. We want safety. That's the big issue. And so they're asking questions, particularly with regard to material science and what it is, what the material is. There's a lot of experience in the body with silk because, of course, silk sutures, which we've been using for hundreds of years, are known to be biocompatible, and I'm pretty sure they have actually sericin in them. So silk sutures have been used for years, but that won't do them. They want more because we've got a new composite, we've changed the silk integrity, and so we need to prove safety to them before they'll allow us. So we're in the midst of that now. It's not years away, it's, I hope, months away, I hope.
So it is possible that within years, this could be a relatively available technique here in Australia?
Once we get FDA approval, we're going to be doing our clinical trial here in Australia. And so we're hoping that will be next year, or starting at the end of the year.
What a journey, like quarter of a century, man.
Oh, honestly.
Quarter of a century.
I've learned a lot about how to try and take an idea from bench to bedside. It's an onerous task.
I've learned so much from you as compared to firstly what I learned in my medical studies, and also that I was possibly pronouncing the word otolaryngology, perhaps with the soft G, not a hard G. This is amazing. Look, how can people follow you in your fine work?
Karl, thank you, first of all, for letting me tell you a little bit about our work on the tympanic membrane. And if people want to find out more, they can go to our website, www.earscience.org.au. And you'll find there a whole lot of interesting stuff, including our work on Cleardrum, tympanic membrane repair.
Well, I'm looking forward to maybe one day having a little drink with an umbrella when you finally get TGA and FDA approval. Thank you so much, Professor Marcus Atlas, for coming on today.
Thanks, Karl. I really enjoyed myself.
Shirtloads of Science is washed, spun and aired by the University of Sydney. I'm Dr. Karl, coming to you from the lands of the Gadigal people of the Eora Nation. I acknowledge Aboriginal and Torres Strait Islander peoples as the first Australians and traditional custodians of the lands where we live, learn and work.